I'm Taylor Heald-Sargent. I work in Dr. Gallagher's lab at the Loyola University Medical Center. So now I'll take these cells, which I already looked at, and I know that they're way too overgrown, so they're like a plant that's too big. And you can also tell because their media, which is the fluid above them, is a nasty yellow color. You like it to be that red that it starts out at. So these were cells taken from an adenocarcinoma of the colon. And since they were cancer cells, they're immortalized, meaning they can just keep dividing and dividing and dividing as long as you want in the lab, which makes them useful to study viruses and other things that need cells to grow on. So what I have to do first is I have to take off this old media, which is basically like the food for the cells. And I want to remove it. It's not good for it anymore. And I need to clean it off so that there's no more left. We just use saline to clean the cells, salt water. Nothing fancy. And now the cells are all attached to the bottom of this flask, so they're all sitting there. And these are pretty adherent cells, so they won't move while I'm putting on the saline. They'll stay there as I wash them. Some cells don't adhere very well, and when you put the saline on, they might fly right off, and you have to be pretty careful. These are robust. So now I'm just washing them, and then I'm going to add a chemical to them. It's actually an enzyme called trypsin, and this trypsin will break some of the bonds that the cells have with the plate. So with the bottom of this flask, and by doing that, it'll make the cells float so that they will no longer be attached, and then I can lift them up and move them into a bigger container. But just like the cells like to grow at 37, this enzyme only works at 37 degrees. So I have to put it back in here for a little bit. And that'll take about 5 minutes. Are lifted up off the bottom of the flask, so they're floating in this very little bit amount of the enzyme. And I don't want the enzyme to be able to chew up the entire cell, so I need to stop the enzyme from working. So just by adding back some of this media, it contains components that will stop the enzyme from working and save the little cells. Now remember, the goal of this was to allow the cells to have some more room to grow. In order to do that, instead of moving it to a bigger flask, I'm actually just going to take out a lot of the cells and throw them away. So now I'm only leaving about 20% of the cells that were originally in there. And I'm going to add back some fresh fruit so that those 20% can grow up in the increased space since I removed so many of the cells. I just have to mark down that I passage them so I can keep track of how many times I do this. They're on passage 75. And then I put them right back at 37 degrees Celsius, 5% carbon dioxide, where they're happy.